We could probably have a half dozen speakers on this subject and not even begin to touch all the issues with it, but the last subject is bacterial regrowth. And we've already heard several times today about sediment harboring bacteria, fish harboring bacteria, and so it's obviously the regrowth and the harboring of bacteria in certain sites is something that we need to be thinking about and working on. And Dr. Peterson from the Soil and Crop Sciences Department at Texas A&M has been looking at the effect of carbon on bacterial regrowth. So with that, I'll turn it over to Dr. Peterson.
Thank you, Dr. Wagner. This is the first time I've ever been to one of these Texas roundtables, and I must admit I've been very, very impressed with all of the presentations. Mine is very short. My background is not E. coli or microbes, but it's more in water quality, water chemistry. And I really only get involved in E. coli when I have a student that insists that they do something with E. coli and water chemistry. So carbon is the topic, dissolved organic carbon. Ultimately, the source of all dissolved organic carbon in streams, natural dissolved organic carbon, is photosynthesis. Thurman, 1986, determined that to classify dissolved organic carbon, it was a range of, or a gradient of molecules that were less than 0.45 microns. So the dissolved is less than 0.45 microns. The particulate is that's retained on a 0.45 micron filter. One of the most important things of dissolved organic carbon is that it's a microbial substrate. It's also important in stream because it protects aquatic fauna from UV light. The bad part of it is a transporter of metals and pesticides. And as mentioned earlier by one of the other speakers, it is implicated in trihalomethane production. Basically, when surface water is extracted for use as a drinking water source, and it's high in dissolved organic carbon, the water is chlorinated before it goes out to distribution points. Trihalomethane can be formed. They're implicated in cancer and also spontaneous abortion. Finally, the loss of carbon from the watershed represents the loss of what I would consider soil carbon. Now, I didn't go into objectives and methods here. Our question was, in terms of E. coli and dissolved organic carbon, really did it matter where the E. coli came from, where the carbon came from in the watershed? So basically what we did, we extracted St. Augustine grass with this side, and this side is live oak leaves at a 1:40 extraction. We autoclave sterilized them at 125 for 1 hour. We then used this source of carbon to get different concentrations. So we see here we have a control. Now this control is pure effluent diluted with ultra-pure water. So the dilute effluent is contributing 2.8 milligrams a litre dissolved organic carbon. We're then going up from 12 up to 100, and here with the grass, 16 to 138. The spike of effluent in each of the incubation flasks was 150 mils. Our ultra-pure water was variable to make up the 500 mil flasks, as were our extract spikes to make up our concentrations. These were incubated at 25 degrees C, and approximately every 6 hours, so we went, whoa, 6, 12, 18, 24, then we went to the 36, 48, 72 hours incubation. During those periods, aliquots were removed, and we used the Mtec method, and I won't go into details on that. Emily gave a very fine talk about it. So it's the Mtec EPA method 1603. We had 3 replicates for every quality. So by quality, we either mean leaf extract or grass extract, and quantity. Plus, we also had to do quite a few dilutions to avoid this overgrowth. We actually did the experiment probably 3 times before we perfected it and knew exactly what our dilutions were to be. So this is based on that final experiment. So in terms of results, control, effluent, E. coli, impure effluent, with or diluted with ultra-pure water will not grow. For the leaf, high concentrations resulted in significantly higher E. coli growth. Lower leaf concentrations, lower. When we look at the grass, though, our highest concentration of 138.2 milligrams DOC really didn't induce a lot of growth at all. So we were really confused about why this would happen. It seemed logical to us that maybe dissolved organic carbon from turf grass would be more palatable, smaller molecules than the dissolved organic carbon we were getting from leaf litter, for example. We're also worried about autoclave sterilizing, because this would have broken up some of those carbon molecules and bonds. So the next slide, we looked at, we normalized to millimoles. We looked at the carbon, so we have a pretty good relationship between the carbon and leaf, between nitrogen and leaf, between orthophosphate and leaf. The grass is coming up pretty well here, but dropping off at these higher carbon, higher nitrogen, and higher phosphorus levels. We then looked at ratios. The CN ratio, again, pretty good relationship with the leaf CN and E. coli. Also, pretty good negative relationship here between the NMP ratio of the leaf and E. coli. With the carbon and phosphorus, the ratio, we were able to get a decent correlation, if you like, between those two and E. coli growth for grass and leaves. So the next thing we looked at was something that I'm more comfortable with, which is biodegradability of dissolved organic carbon. So what we have here are our controls, our low concentrations, which were those at about 10 to 12 milligrams a litre, the medium concentrations, and those concentrations that were 100 plus. The black should be the leaf, and the white the grass. And we were seeing between 60 and 80% of that carbon was being degraded or utilized by E. coli. Similarly, for total nitrogen, we were seeing up to 80% being utilized. Phosphorus, however, 40 to 50%. So phosphorus, we felt, wasn't limiting. So what we have to start thinking about with E. coli regrowth is what's limiting it, and how do we limit it even further to stop that regrowth in the watershed. What I'm going to just, sorry about this. This is something you see quite often, certainly in our region. People will edge the grass, they'll leave it, irrigation event comes along, a rain event comes along, and this is all washed into the stream. It's a carbon source. So one of the BMPs, really, to my mind, is make sure everybody is cleaning up after mowing. So finally, we looked at the carbon to nitrogen ratio divided by orthophosphate, and we found a really good relationship between the grass and with the leaves using this. So as that CNP ratio increased, so did E. coli. This is all very well in a flask in a laboratory, but how does it work in real life, in a real watershed? This first slide, these are independent streams in the Bryan College Station region. The first slide, these are all downstream of wastewater treatment plants. So the wastewater treatment plants, they don't produce an awful lot of carbon, as we saw in that earlier slide with the concentration about 2.8 milligrams a litre. But they do produce an awful lot of phosphorus and nitrogen. So that's what's bringing this CNP ratio down. But we see a really good relationship, and this is in the stream, the CNP ratio compared to log E. coli. This second set are streams that do not have a wastewater treatment plant in the watershed. And overall, we see as this CNP ratio increases, so does E. coli. But what's happening here? These four watersheds, they're all urban watersheds, and they have more than 35% of their urban area under turf grass. So that's either golf courses, city parks, neighborhood grass or turf grass. And these have less than 35% of their total urban area under turf grass. So it would seem then that turf grass could be something that we should really look out for, and certainly we should be looking out for clearing up after mowing. The second part of our question really was, well, where is all of the carbon coming from in a watershed? So we developed a method using diffuse reflectance near-infrared spectroscopy to actually find signatures of our watershed sources of organics. The first slide, this is St. Augustine grass, riparian vegetation, post oak, effluent, impervious runoff from soil, and these are ultra-pure water blanks. So using cluster analysis, we were able to cluster these different sources of carbon in a watershed. From there, we sampled 4 local creeks over a year, used our evaporation method, scanned them with NIR spectroscopy to find out where they actually fell in here. And this data here is telling us a percentage of streams, for example, this one, Carter's Creek, in the winter, most of that carbon is coming directly from soils, some is coming from post oak litter, a certain amount is coming from impervious surface runoff. Carter Creek has a source of effluent, but in the winter and the summer, in the winter probably dilution with rainwater, in the summer probably dilution with irrigation water, the signature of effluent disappears. We're getting effluent though in Hudson Creek that has no wastewater treatment plant, Wixon Creek, no wastewater treatment plant, and Wolf Pen Creek, no wastewater treatment plant. This is really very preliminary data that we have here. Once I get back to the lab, we intend, instead of looking at feces for E. coli, we're going to look at feces for carbon signatures based on different diets. So with the cliff swallows underneath bridges, I've allowed them to nest by my house. I've been daily collecting their feces, so I have a source there. Anybody that has chickens, bring me some of their feces. I've been in the woods at the back looking for deer feces. My students have a big bag called Jacqui's poop. So we hope that we can develop this an awful lot more. If it is successful in terms of being able to tell you where your carbon sources are from your watersheds, I think it will be an awesome tool. It's cheap. It won't cost anything. Really, we just scan it, get the signature, and as we get this source database bigger, we should be able to give you the dominant source. And then hopefully we also want to mix sources so that we can give percentages of sources from different whole watershed regions. So final slide. My thoughts on best management practices is to keep our carbon and nitrogen on the landscape. Run on, not off. Don't over-irrigate, and I'm probably preaching to the converted here. Rain harvesting, keep the water on your land. Rain gardens, wet cells, detention ponds, grassy swells, and avoid vegetation getting into those road gutters and storm drains. If we can keep the water on the landscape, give it a chance to infiltrate before it gets into our streams and rivers, I think we're onto a better chance. Thank you. Questions?
I've got one. I was just, you showed some correlations there between the dissolved organic carbon and the E. coli. And I guess I was wondering what your thoughts were on, is that a result of those coming from similar sources? You know, if it's coming from feces, there'll be a lot of carbon associated with that, or is it because having the presence of the organic carbon there is providing a food source and thus encouraging growth or maybe increases in
the presence of having the carbon is a food source. It is a substrate. But what we've also got to realise is, we can have enough protein, but if we haven't got the starch, for example, our potatoes and vegetables, we're not getting a decent diet. So for me, the take-home message here is, yes, carbon is a substrate, but we also need those relative proportions of nitrogen and orthophosphorus so that the E. coli will grow. Those particular correlations, the best ones were with leaf litter that had a higher CNP ratio. The grass, it was limiting in carbon and nitrogen. Yes.
If you would go back to your slide for the 4 watersheds, please, yeah.
This one?
I'm a little bit familiar with the Wolfman Creek there.
Yes.
And so the contribution from the impervious surface in the summer and then in the fall, there's a very different story there. Can you kind of guess on that?
We finished taking those samples this year in the spring, so those fall would have been last fall. I don't think we had an awful lot of rain. I think it was somewhere in the region of 145 millimeters in the fall. We did have more rainfall in the summer, actually, so I think it was 168 or something, maybe 20 millimeters more. But we've also got that irrigation water coming in. Yes.
Okay, thanks.
Would total organic carbon instead of dissolved provide a similar indicator?
Can you repeat the last thing? I didn't hear it.
Would total organic carbon give a similar indicator as would dissolved organic carbon? We tend to split it, and the reason we split it is that we have 400 ppm bicarbonate in our municipal tap waters, which is also contributing to very high bicarbonate in our streams. We do also see a very good relationship between the dissolved organic carbon and the bicarbonate. We're still doing tests on that. We're not sure whether bicarbonate is competing in the soil in terms of anion exchange sites. So in answer to your question, the fact that they are very well correlated, I haven't looked at the data, so I can't say definitely that yes, we would see a similar result with bicarbonate, but possibly. Yes?
There's been a lot of work on organic matter in rivers and streams. So I was kind of really curious about your leachate experiments. In terms of, did you use senesced grass and leaves?
It was fresh.
Or were they green?
They were green but dried. We allowed them to dry for a week before we extracted. Typically, if you let anything dry, when you're looking at carbon, you're going to get a huge flush. And our idea was to get the high concentration number so we could bring it to the highest number we could get and do a range.
But in terms of organic matter contributions to the streams, most trees and grasses suck up all the goodies before they drop the leaves. So the leachate is different for green leaf versus a senescent.
Yes, it is. We can get into a different subject here because what we are also finding in our region, we have very high sodium irrigation water, high sodium in the streams. And we have a paper in review in biogeochemistry now looking at different types of leaf litter with different sodium absorption ratios, sodium concentration, exchangeable sodium, and we're finding that the higher sodium, the more carbon is pulled off. So in terms of a watershed scale landscape in our region, or any region that has relatively high sodium, and this is going to be important when we get to recycled water, which is going to be much higher in sodium, that we'll see a much higher release of that carbon, be it fresh or senescent.
Okay, and have you factored in the fact that there's such a broad range of decay characteristics of different types of leaves?
In terms of that particular paper, yes, we have different solubilities.
No, decay, decomposition.
Oh.
Some leaves take 3 to 4 years to decay. Others can decay inside of 2 or 3 months.
And when we talk about buried bag studies, that's been shown. When we put leaves in stream water, we found certain amounts of carbon coming in terms of rates of decomposition. That's something we've actually talked about going further, doing a buried bag type situation in the stream. Any other questions? Would you?
Thank you. I think the limiting nutrient aspect of that was really a concept I've never thought of or applied to bacteria. You know, we've used that for years in the water quality in relation to algae and other things like that, but you know, they're all primary producers, so it makes sense that that's another thing that another area that we could apply the limiting nutrient aspect or concept to.